Hello, everybody. Welcome, and thank you for joining us today for Noah Kolander's Thesis defense. Yeah, we are excited. So I think some people are watching online, and so if they have any questions or anything, afterwards put it in the chat, and we will try to ask Noah. We'll try to make him squirm a little bit after he defends his thesis. And then after it's done, his committee, we're gonna whisk him away for a few minutes to torture him a little bit more, and then we'll bring him back, and we will all celebrate. So yeah, I'd like to thank everyone for coming. Noah's committee, so Maya DeVries, his thesis advisor from San Jose State. I'm his co-advisor here at Moss Landing Marine Labs, Scott Hamilton, and then Luke Gardner. And Moss Landing Marine Labs is also his. He's sitting over there, is also his on his thesis committee. So yeah, thank you everyone. So I'll hand it off to Maya. She's going to start off a little intro for Noah, and we're going to tag team this one a little bit.
Yeah, hello everyone. Thank you so much for coming. We are super excited for Noah's defense. Yes, okay. Oh, great. Yeah, and this is my first Moss Landing student, so I'm also super thrilled. Yes.
Where do you go from here?
I don't know. I got two sitting in the crowd, so big shoes. Yeah, we're going up. So this is Noah growing up at the beach. You can tell that his family always loved going to the beach in Southern California. Noah is one of 4 brothers, and as a fellow boy mom, I know how fun it is to have lots of boys. And he was the 3rd, so he's like the 2nd smallest in the photo there. And
I'm the biggest now.
Noah was also always a budding young scientist, where he had many presentations, poster presentations throughout his career, his young career in elementary school and then also college. He went to Concordia University and studied with Sean Bignami, and he did abalone aquaculture there as well. So after undergrad, he actually worked as a Florida agriculture inspector, and he was a lab scientist for COVID and STD testing. But you can, and
Got any weird diseases?
yeah.
Keep them going.
And I actually remember this from when we first spoke over Zoom. You were like, well, I'm in Florida now, and I am testing for COVID. And I was like, well, that's very useful. So useful skills already. But you can tell that he was super active at Concordia University, already starting to do aquaculture and inspiring young scientists, and already being super helpful because he also was the lab manager for 2 years at the marine lab there that they were just starting. So all of these themes carry through his career as well. Noah loves inverts. As you can see, many photos with fun invertebrates, and from the time he was a child until now. But Noah...
But he hates fish. He's also in the fish lab, but he has a lot of animosity towards things with gills for some reason. All right, Noah came to Moss Landing Marine Labs, and he quickly jumped into basically anything aquaculture, and he's quickly really become an expert at doing really anything you need. So, he can plumb it, he can fabricate it, grow it, spawn it, cook it, basically anything. And he's really become indispensable to many of us that are working on a whole variety of aquaculture projects at the lab, from seaweeds to inverts to even fish. Yeah. All right, if you look back, Noah's also really innovative. He likes to invent things and come up with new crazy designs. He's pioneered new ways of cleaning tanks. You just get into them. We went down to Baja, and he brought down some microalgae cultures in some Coke bottles, and that was apparently the key to getting him to survive for 3 days on the road. He brews lots of interesting stuff as well. Again, he's really innovative, but he's got this weird fetish for Home Depot buckets. What is happening on the left there? Some sort of a dairy farm? Amazing. And some other of Noah's inventions involve clothing. Here he's developing new biomaterials for sustainable bikinis. These ones reminded me of Inspector Gadget with those long go-go gadget arms. But, unlike Inspector Gadget, whose most of his gadgets fail, most of Noah's, they work. They do, Yeah. Noah's been a real indispensable member of the Moss Landing community for the last couple of years. We won't say how long. But he's jumped into so many different projects. Any opportunity, if you can get in the water, on the water, under the water. He's helped with the Surf Zone project. He's helped so many students with diving-related projects. He loves to do Halloween. He's presented talks at many different conferences. He won the best student talk at the Western Society of Naturalists in 2023. He got the runner-up the next year after that. He's presented a couple times at Aquaculture America. And, he loves to have fun. He smiles. He makes all of us smile. Look at those eyes. Lab Olympics. Third place forever, I guess. You're the third brother in line, always third place. Yeah.
All right. Technically, Noah's also at San Jose State with me, where my lab is, and he's also indispensable there. He did not spend as much time in my lab, of course, as here. But that is him working on the Instron, which you all will learn about, and giving a poster at San Jose State as well. I also wanted to just jump in with how helpful Noah is all the time. I remember, and just innovative, I remember, I think you had only been here a week, and I came back down to visit here, and you had already learned how all the flow meters worked on all the tanks, and you were already checking them and troubleshooting things, and you'd only been here a week. So that is one of the reasons why Noah got to come to Maui with me and my lab in 2023. Yeah, 2023, for sure. And obviously he was super helpful while we were diving, but also incredibly helpful and fun when we were touristing around Maui because we couldn't dive in the afternoon. So we have lots of fun photos together. Also, Stephanie came to visit, which was lovely. And my kids were there. My older one loved Noah, and you can see they were about to go whale watching, and then he also just never wanted to let go of Noah's hand. So inspiring little guys, but also big kids and undergrads. Noah inspires many young scientists, and he, along with Kirsten Thigpen, are our first and only Aquanaut mentors officially, and they have stewarded 2 amazing cohorts of undergrads through learning aquaculture, and also you've helped with my marine ecology classes when we've come down to count snails, which has been amazing. And of course, not just humans, he also inspires his own dog baby Moose.
Noah's also in the Baja class that we did last spring, and he was, again, an valuable member of that class as well. So he drove the truck and trailering all the way down. I always offered, and he said, no, I will be the driver. I don't trust you. The van got stuck multiple times in the sand. Not by him, but he was the first one to go dig it out, and he also would not let anyone else help dig it out because only he could do it right. For this class, everyone has to design an independent project, and Noah had one that was basically 2 master's theses that he was going to do in about 2 weeks' time with 10 different objectives. But he actually did almost all of it in 2 weeks, so I think he won the Guinness Book of World Records, again, for most science projects completed in a 2-week period. My family was also there, and my daughter was always looking over his shoulder. And again, he was so helpful to her and kind of explaining and teaching her what he was doing. He even let her paint his nails pink. There he's showing them off underwater. And Noah he has a big heart, as you can see. And one final message, and that's that. All right, and with that, we're going to bring Noah up to present his thesis. Sorry, Mom.
On to snails. I mean, I have this slide again at the end, but just to thank every one of my classmates, friends, family that helped me through everything. Some great pictures up there. I'll throw it up at the end and just let everyone stew on it. But before we get to that, we got to get through snails and seaweed. So yeah, thank you all for being here today. It's exciting to see everyone. Today I will be presenting my research that I've been working on for the past year or so. Abalone and seaweed co-culture, growth and shell biomineralization of an iconic California gastropod. So as I think we're all at least semi-aware of, I talk about fish. I even have a picture of one in my slide. But as we have been growing as people for like thousands and thousands and thousands of years, millions of years, we have been advancing technology and we have been eating foods, and a lot of time those things go hand in hand. This is really great for us. It's allowed us to sustain populations that we have today. But on the other side of that, it's been really detrimental for some of our favorite foods. Shown here, we have a scuba diver going down in this super high-tech gear, collecting abalone, whereas once you had to dive down on one breath, maybe grab one, this guy can go and just pull them off the rocks. Up in that top right-hand corner, we have a trawl net that in one fell swoop can get thousands, if not millions of fish. So as our skills have increased, unfortunately, the wild stocks that we're taking from are not doing too hot. And this, of course, leads to losses in fish in general, and then even closures in those fisheries that we're seeing, where it goes from, on that left side there, we have a tiny little bit of overfishing, and now we get to the unsustainable point on that right side there. So on top of this, we have numbers that are getting decreased, and then we have the act of illegal fishing that takes those numbers, those animals that are already at these low densities, and then you take more animals out of the population, really just like driving it home, not great for them. But fortunately, we have aquaculture. If you're not familiar, aquaculture, raising these animals in seawater systems just like a farm. And this has been on the rise over the last several decades. In this graph here, we have fish take, and so we can see that in the blue there is our capture fisheries, and this has kind of gone up, plateaued, wavering a little bit, but that orange line that's been steadily creeping up and now finally exceeds the take in wild fisher captures, which is huge for restoration of species and just another avenue in order to get animals to feed ourselves. Unfortunately, we have this issue of climate change, which I think we're all pretty aware of. And on top of that, or more specifically, what we'll talk about is ocean acidification. That small avenue of it. Ocean acidification occurs when we have large amounts of CO2 released into the atmosphere, and this in turn then diffuses into the water. Now this causes a host of problems, all stemming from this area where we have more CO2 in the water, and this causes our pH of the water and our carbonate concentrations in the water to decrease. We already see negative effects from this in animal reproduction development, in their growth, in their survivorship, and their calcification rate, which is what we'll mainly focus on today. So up here we have a nice little baby oyster on our left side that was raised in a pH of 8, and you can see it's pretty symmetrical, it's a nice looking baby oyster. And then on our right side, we have an oyster that was raised in a pH of 7.49, and you can see just that little decrease in pH there has some pretty negative impacts on it, where it's got that huge divot in the side and all these bumps and lumps around it. Not looking great. So to get at what the ocean acidification is really affecting our calcifiers in the water, we can see this equation here, and it looks big and scary, but really it's all just kind of moving back and forth. On our left side here, we have CO2 in the air, and that double arrow means that it can kind of bounce back and forth between these two states, depending on where it's needed. And so the CO2 goes into the water, and then that reacts with the water, and then we create carbonic acid and bicarbonate and carbonate. And all the meanwhile, we have these little guys popping up, those H+, and those are hydrogen ions, and this is what causes our seawater to get more acidic. And on the end there where we see our 2H plus the carbonate concentration, this gets tricky because sometimes our equation needs to be satiated, and if there's not enough carbonate in the water itself, the water can pull this from other organisms that have calcium incorporated into their bodies. So at a pH of 8.1, this equation is pretty well balanced. Here on this graph, we have concentration on the y-axis and then pH on the x. And that black dashed line there is showing our current ocean pH at 8.1. And as our CO2, it's kind of like a yellowy line there. As that line increases, we see decreases in all these others. And with our projected ocean pH decreasing, we can see that our carbonate level is decreased. Our pH there on the x-axis is also decreasing, which is not great for our calcifying organisms. So our carbonates, as I've kind of said before, are calcifiers. They produce these shells made of calcium carbonate. And up here on the top, we have a shell that has been in a pH of 7.7. We can see this deterioration happening. And at the bottom, we have a pretty shapely shell from a pH of 8.0. And not only does ocean acidification cause these shells to degrade, but it also causes them to lose some of their form and their function as animals growing in the wild. And this is super important because not only does it affect these calcifying animals, but it also affects other animals in the ocean. Fish have calcifying parts. There are some algae that use calcium to build their bodies. So it doesn't just affect our snails. And so when there's these limitations to calcium carbonate in the water, sometimes they have to shift around and reallocate resources, and they include other minerals into their shells, but we don't know how this is really going to impact them in the long term. One such animal, this calcifier, is a red abalone, and this will be the study species for this talk. Abalone, if you're unfamiliar, are a marine gastropod. They're an ocean snail. And they grow their shells their entire life. When they're tiny babies and this big, they've grown that shell, and when they get to their maximum size, being like a foot long, the biggest one they found, they've grown that shell their entire life that can range upwards of 50 years in the wild. So a relatively long-lived snail. And because of their large size, they've been highly coveted by people that live throughout the lands. Historically, the Rumsen and Ohlone people that lived in this area utilized them. And you can still find all over the greater Monterey area these piles of shells, these middens, where people have taken these shells because the abalone provides so much meat and utility. So they have big cultural significance, both as food, and then up in Northern California, today even, there's a pretty big historical, big nostalgia around abalone. And so in this picture here, right in the middle with all those pillars shooting up from their front yard, this person has become so attached to their abalone. On all of those pillars, those tiny little plate-shaped things are all abalone shells. And so they've taken all of their abalone that they've caught over the years and tacked them up onto these poles. It's pretty impressive, although if you're a little abalone, I imagine it's kind of a horror scene crawling down the streets. So on top of the utility of their food that provides us with, there's also some pretty fantastic artwork that's been made from them. I have this abalone bolo tie, my abalone belt buckle, abalone wedding ring. Steph has some abalone stuff as well. It makes for really pretty decorations and really amazing art too. Their nacreous layer has this really nice iridescence to it. You can kind of see the different layering of it too. And now sold online, you can find stuff from anywhere from a couple of dollars to just a little abalone shell to over thousands of dollars for these really intricate pieces of work. But unfortunately, as I was kind of talking about earlier, not everything is looking too hot for our animals that have this high value to them. In this picture here, we see some classy businessmen standing over a literal mountain of abalone shells. You can tell it's not great for their populations. And now this intense take and honestly our love for the animal and the love for the money that the animal brings in has caused some pretty steep declines in their populations, both commercially and recreationally. These animals were fished for thousands of years, but then really intensely in the last several decades. Here on this graph, you can see landings in millions of pounds. And you can see the different color coordinated for the different species of abalone. We can see a big peak in the red abalone take, and then pink abalone, white abalone, and then they all kind of peter off in 1997, which led to the closure of the commercial fishery, which then eventually followed up with the closure of the recreational fishery in 2018. I'd just like to throw one more thing onto it. There's also this problem of withering syndrome, which is a disease that these abalone can contract that literally just causes them to shrivel up and die. So that's not great. And then as we're destroying these species, taking them out of the wild, we are also doing restoration, which is fantastic. One of the species that has been restored at the California coast is the otter. And this is really good for healthy ecosystems and kelp forests. However, abalone are like a favorite treat of the otters, and so even with something good, once again, our abalone are kind of got the short stick. Fortunately for abalone lovers, you can still find abalone through aquaculture. And there are still farms that operate and sell these to the public. but once again, unfortunately, due to lower production costs outside of the United States and several factors that make abalone quite difficult to grow, the number of farms has decreased from over 24 to now only 2 operating farms in California that are commercially selling red abalone. And aside from keeping abalone in our minds and on our plates, these farms are a really valuable resource. Right now in our aquarium room, Kate from Monterey Abalone Company is doing a red abalone spawn, and so she's producing millions of tiny little baby abalone that they graciously let us do research on. And the abalone that I use in mine were a little bit larger, and so we have like all these abalone from all different life stages where we can see how different impacts like ocean acidification, for example, are affecting their growth and development, and how we can in turn help the abalone industry increase their growth rates. A few other things that make abalone aquaculture difficult. The first one is the spawn to market time. Abalone are a very slow grown individual. These little guys circled here about 3 millimeters long, and they still have 3 to 4 years before they will get to a market size of, and that's only like 3 or 4 inches. So very slow to not a very big size. And then in that entire 3 to 4 year period, you have to keep the lights on, you have to keep the water going, you have to pay your people, you have to siphon tanks. And there's lots of operating costs that are associated with it, in addition to the permitting costs, different various commissions and departments that you have to deal with in order to grow your animals. And of course, we have to feed our animals. And with millions and millions or hundreds of thousands of tiny little mouths, this requires a lot of food. And the 2 farms in California either grow their algae on site, or they'll go out into the wild kelp forests and pull off like the top few feet of kelp off of there. Kelp grows really fast, it comes right back, it's not a big problem. But year over year, it's not always plentiful, and they can't always get it. So on the other side of that, growing your kelp or growing some algae in a farm to feed to them, this is done at those farms, but it's not always guaranteed that you're going to have great production. You have to keep those tanks going, and you also have to have the space available for them. So even feeding the abalone is difficult. And then like the last thing just to throw on the pile is the issue of nearshore ocean acidification. So naturally, the problem with all of this appears to be like stemming from, and definitely the base for everything, is increased CO2 in the system. So naturally, the solution is removing the CO2. That's my talk. So I propose we do this, or we're going to do this with the use of seaweed, this biologically kind of filter that removes carbon dioxide from the system. And we're doing this in the context of integrated multi-trophic aquaculture, IMTA. And so in our IMTA system, we grow dulse, this species of red algae. And dulse is high in protein. It's grown on other abalone farms. They feed it to their abalone. It's obviously palatable to the abalone. It reproduces via fragmentation. So if you have 1 clump, rip it in half, now you have 2 clumps, and they grow up, rip them in half, now you have 4. And so really easy to grow, really easy to culture in tumble culture. And most importantly, at least for the integrated multi-trophic aquaculture part, it photosynthesizes. Now, if you're unfamiliar with photosynthesis, photosynthesis is going to increase oxygen in the system and increase pH while driving the nutrient content down, which creates a pretty happy environment for our little red abalone, which in turn, through respiration and effluent production, are going to decrease oxygen, decrease pH while increasing nutrients, which you can kind of see it makes this nice little cycle where both of these organisms can grow together and directly fuel one another. Now, I'm not the first person to come up with this system. It's been done for a few decades now. Back in 2004, they looked into how the nutrient exchange worked between the seawater nutrients going into the dulse, going into the abalone, and then back into the seaweed. Fast forward a couple decades, and we have our very own Hamilton et al. He does snail stuff too, so it's not just me out of bounds. And he looked at how different exchange rates between a seaweed tank and an abalone tank can affect their growth. Now we know there's lots of questions around abalone IMTA specifically. We can adjust light, we can adjust nutrients, but there are still many other questions to ask. So in the context of ocean acidification, the specific objectives of this project were to determine how variable pH levels affect abalone growth, shell biomechanics, and dulse growth within the context of integrated multi-trophic aquaculture. I hypothesized that abalone grown in this high pH water will grow faster, have thicker shells, develop stronger shells, have a lower inclusion of magnesium in their shell, and show less corrosion overall. and that dulse in those systems at a lower pH will be more abundant. So I'm briefly going to just walk you through how I'm going to talk about all my things, because I did a lot of different things once again. So it gets kind of confusing, but I'm going to try to simplify it. First, I'm going to talk about my experimental design and what we did, how we did everything, and then I'll talk about the dulse growth and the results from that. And then I'll walk you through the abalone growth, the methods and results, and then we'll look at shell composition methods and results. And I'll give a brief summary of everything, and then some conclusions and closing remarks. So at our beautiful shore lab that's just right down the road over there, we have this nice array of tanks here, these 900-liter tanks filled with seawater and 10 kilograms of dulse. And then we threw in 620 red abalone into the tank. And then through this vigorous bottom-supplied aeration, this causes our dulse to tumble around, or abalone can feed on it and get nice and big, and then they poop and pee in the water, and our dulse gets nice and big. So I had 16 of these tanks with 10 kilos of dulse and 620 abalone in them, and each of them were randomly assigned a treatment, either high, ambient, medium, or low pH. And in any of those tanks that we needed to decrease the pH, we did so by bubbling in CO2, because back to this graph here, the inclusion of CO2 drives down our pH. I didn't just randomly come up with these pH treatments. For each of them, I looked at specific tanks. And so for our high pH treatment, I calculated this, or made this graph based on 10 kilos of dulse thrown into a tank. I monitored it for 2 weeks and then took the average of all these different points. And that's our high pH treatment there. In our ambient pH treatment, I did a similar thing, but this time no dulse in the tank, just water going in there. And so we can see there's, just with water in the tank, quite a bit of fluctuation, thanks to our seawater intake being right at a weird level where we get some pretty odd fluctuations, both tidally and being at the head of the Monterey Submarine Canyon. It gets a little funky. So I wanted to kind of plot these so that they would be as close to what we're actually seeing down at our shore lab. Next, looking at our medium pH treatment, this was based off of the high pH treatment, but reduced by 0.3 pH units to simulate the 100 years forecast for what our ocean pH will be. And then in the low pH, we took our ambient pH and dropped it down by 0.3 units, again, to simulate the future ocean acidification. So once we had everything established, daily I would take regular pH measurements to ensure that all of these different treatments are within the bounds that they're supposed to be. And then monthly, we would measure 1 to 200 abalone from each tank, looking at their length, width, and weight. And then every other week, I would net all the dulse out of the tanks, weigh it, and then if it was ever above or below 10 kilos, I would add or remove dulse from it to keep it right at that 10 kilo mark. Okay, so let's get into some dulse results. This first graph here is kind of all over the place. There was a lot of variation among the treatments, and then also a lot of variation, as you can see, over time with their, like, going from our summery months into our wintry months, seeing a decrease in growth. And so this is the first graph that I saw, and it just wasn't as clear-cut as I thought it was going to be. So I wanted to figure out what was causing these differences. And to do this, I put all these numbers into a principal components analysis, where all of these different factors, the PCA groups them and puts them in levels of like relatedness to one another. And so here in this black line, we have dulse growth in kilograms. And then our other two metrics that are pointed in the same direction there are PAR, which is photosynthetically active radiation, and then daylight, and that's minutes of total daylight. And so we can see our dulse is pointing this way, our PAR and our daylight are also pointing in the same direction, which implies a positive correlation, and then they're pretty closely spaced to one another, which implies a strong correlation. So it kind of makes sense. You need sunlight in order for things to photosynthesize. And then tipping the scales the other way, on the back side of this, everything that's pointed in the opposite direction, this is negatively correlated. So as one value is going up, the other is going down. And almost at 180 degrees from our dulse growth, we can see pH. This is a really big driver of our dulse's growth. With that decreasing pH, we see an increase in dulse growth. So for each of the treatments, I have one of these principal components analyses, and I've highlighted what I want you to look at, when I want you to look at it. So in this first circle here, we have the dulse growth for each of those different treatments. And highly correlated with it, we find our minutes of daylight and our photosynthetically active radiation, which I think we would all statistically makes sense. On the opposite side of that, we see, so negatively correlated with our dulse growth, we have temperature of the tanks. Dulse is a temperate species. It needs cold water to grow. And so we can interpret this as either if the temperature is increasing, our dulse growth is decreasing, and flip-flop it. If our temperature is decreasing, maybe our dulse growth is increasing, as they're oppositely correlated from one another. The next big negative correlation that we see is abalone length, which makes sense. This is a huge driver in those tanks. As our abalone got larger, they were eating more food, and so we saw less overall dulse in the tank. And I'm pretty sure it didn't go the other way. And then finally, we look at the raw seawater pH, and we can see again that this is a really big driver for these systems. As our raw seawater pH was decreasing, we saw more overall dulse growth. So now to kind of like synthesize everything back, I took all of that data and mean zero transformed it. So everything is kind of related to itself in this week over week. And we can see here that there are, on average, there was more dulse produced in our low pH treatment compared to our high pH treatment. Next, we'll get on to our abalone growth and results. I'd like to take this time as well to thank my dog Moose. She has been a great supporter and supervisor in all the work that I've done here. Anyways, on each of these result slides, I'll orient you. In the bottom right-hand corner, there'll be a picture of the abalone showing what exactly we measured. And then up above the graph here, which I probably should have explained on the last slide, but I didn't, these letters above each of these bars indicate significance or like relatedness to one another. So any two bars that share a letter are statistically similar to one another, and any that have different letters over them are statistically different from one another. In this graph, on the y-axis, we have growth per day, and that's where all of my metrics that I'm looking at will be on that vertical axis. And then on the x-axis, this should pretty much be the same for all of them, where those different pH treatments are labeled and color coordinated throughout. So what did we see? You've had a minute to take it all in. When we look at growth per day in micrometers per day, we see that abalone raised in our high and ambient pH treatments on average grew larger than those raised in the medium and low. However, our ambient treatment grew statistically faster compared to our low pH treatment. And just to preface kind of all of this data moving forward, abalone are notorious for having a pretty wide range of growth. You start all your abalone at a really small size together, all within a few millimeters of one another, and then after 5 months, you can see that spread just gets really, really wide. And so in some of these graphs that I show, it looks like there should be differences between stuff, but it's just, there's so much variation between them, and the stats don't lie. And so to capture some of this variability, we wanted to look at the top 25% of the abalone growth in the tank. And so we took our top 25% performers from each of these different treatments. Kind of look at the maximum potential growth. And so here we see that abalone raised in our high and ambient pH treatments were larger than those medium and low, and both our high and ambient pH treatments were significantly larger than our low pH treatment. In this ridgeline plot here, again, we can see that distribution of all the different sizes between the abalone. So it wasn't just one treatment that was really spread out all over the place. And those dashed lines there represent the different means for those different treatments with ambient, high, medium, and then low pH treatments. Next, when we look at shell area, here, even though the high and ambient pH treatments were on average larger than the medium and low, there was no statistical difference. Although our p-values were pretty close, but not quite under that 0.05. But when we take our top 25% of our growers, we see that abalone in that high and ambient pH treatments were significantly larger than our low pH treatment. And then when we look at weight of our abalone, which is a good metric to have, that's the foot, that's the part that people are eating, one of the big parts that they care about. And in here, we see that abalone raised in the high and ambient pH treatments are significantly heavier than those in the medium and low pH treatments. And next, we kind of took a more holistic look at our abalone, taking a body condition metric, which we have defined as weight per millimeter squared of the shell. And in here, we see that those abalone in our high pH treatment, not only on average were larger than any of the others, but significantly had a better body condition than our medium and low pH treatments. And again, we can see that in this ridgeline plot with a big spread over everything, but our high and ambient pH treatments ahead of our medium and low. So after we looked at all of our growth throughout the project, we then had some shells to look at. And so I kind of want to orient you with the abalone shell. This is the underside of the abalone shell that has this beautiful iridescence. Abalone shells are made up of calcium carbonate, but they have two different crystalline forms of calcium carbonate. So they have calcite and they have aragonite. And when we look at these shells from a side profile, we can clearly see the defining line between these two crystalline forms. And when we look at them under the scanning electron microscope, it gets even cooler, and we can really see the crystalline structure between our calcite that's kind of amorphous all over the place, and then our aragonite that's like super particular, and it's like type A versus type B. This is very type A, not me, accretions of the calcite or aragonite. Now on the bottom side of the shell, where we have all this beautiful iridescence, that is the nacreous layer, and this is where it's already been deposited. That kind of blurred, opaque, translucent edge to it, that is where new shell is being accreted. And so what that looks like when you zoom in 2,500 times are all these tiny little platelets, all these tiny pancake-looking things that will eventually grow up, and you can kind of imagine if you turn those pancakes on their side, they would have that flat look to them there. So now we know all the different parts of the abalone shell. There are lots of tests that we can do with the abalone shells to see how the treatments that they were in during their life impacted their shell growth. The first of which was testing the strength of their shells, which is pretty relevant for commercial and restoration aquaculture. And to do that, we used this big, beefy Instron device, a little larger than the one that Maya has at her lab. It was ridiculous. It's literally like metal I-beams. They built it themselves, believe it or not. You can't buy this from Fisher. And then it has a hydraulic press on the top that pushes down, and then a force sensor on the bottom. So what that looks like is, this is a video, by the way, so you gotta watch. And as our force increases, eventually we get to the point where the shell can't take it anymore, pop, and it cracks. And then Rachel Crane at Hopkins Marine Station, I mean, she helped build that system and did all the R coding for everything, helped us with all the sampling. Thank you very much for all the help. And so she developed this code that spits out then a value to give us the force required to fracture these shells. We also, kind of in the same vein, looked at shell thickness, and then the individual thicknesses of calcite and aragonite in the shell. And to do that, we would take our shell, cross-section it, polish it, and then look at it under a stereo microscope. We also wanted to see what the shells were made of and what minerals they were incorporating into their shells. Now to do this, we used an X-ray fluorescence detector, or XRF. There it is, pictured, nice little compact handheld unit. And with this, we looked at both the dorsal side of the shell, the back side, and the underside, where all that nacreous, pretty iridescence is. I guess I should say how it operates. So with this machine specifically, it shoots X-rays at your sample, which then causes other X-rays to be reflected off of your sample, and then it's a really crazy amount of computing that goes into it. And then it quantifies these different characteristic X-rays that are coming off of it as elements. And so it literally tells you how much calcium, how much everything is incorporated into the shell. We then took those shells and put them into the scanning electron microscope, where we got those really cool pictures. And here we looked at how those nacreous platelets, those little pancakes, were being affected by ocean acidification. And I asked 25 volunteers, I trained them on what a good nacre platelet looks like and what a bad one looks like, and then asked them to rank them on a scale of 1 to 5. Thank you to all my volunteers who helped me with that. I know it wasn't a small amount of work. And then while they're in the scanning electron microscope, there's also an elemental analyzer aspect to that, and so we're like, why not do 2 different ways to look at the same thing? So we use energy dispersive X-ray spectroscopy, or EDX, to look at the elemental composition of the shell in several different locations. It wasn't exactly the same, but we still did look at both the ventral side, the nacreous side, and then the dorsal side of the shell, so that we could compare them and see if maybe one is better than the other. So what we saw after smushing a bunch of shells was that abalone raised in our high pH treatment, on average, took more force to fracture than any other treatment, and the high and ambient took significantly more force to fracture than our medium and low pH treatments. And then when we normalized that for shell length and in shell area, we saw similar trends where the high and ambient pH took more force to fracture than our medium and low. When we looked at shell thickness, just measuring these as calipers, again, our high and ambient kind of reflecting what we saw in the force required to fracture the shells, that our high and ambient pH treatments were thicker than our medium and low. And then to kind of, once again, take a more holistic look of the size of the shell and the thickness of the shell with the thickness to length ratio. Here we see that those abalone raised in the high pH treatment had a significantly greater value than any of the other treatments. When I looked at calcite thickness, although on average it was greater in the high pH treatment, once again thinking back to all the variability behind everything, statistically it was not statistically different from our ambient, medium, and low, although the p-value was 0.1, so it's like right on the edge there. You can do with that what you will. However, when we looked at the aragonite thickness, we didn't see any statistical difference, although the ambient one in this was thicker than the others. Next, when we look at our XRF, magnesium to calcium ratio, here we saw the opposite of what we hypothesized. We thought that there would be more magnesium in our low pH treatment, but we actually saw more magnesium in our high pH treatment, and not even just like the average is a little higher, but it was significantly greater amount than any of our other pH treatments. And then using the EDX2, which is really nice that the results kind of backed one another up and it wasn't like flip-flopped, have to throw one out. But once again, we see a greater inclusion of magnesium in our high pH treatments compared to the others. So then when we compared those two to see if there was an effect of different analysis techniques or the different sides of the shells or among the treatments, and here we did not see, again, the p-value is 0.09, it's right on the edge there, but technically not statistically different from one another. So you can use either one of these analyses on your abalone shells at least. When we look at the scanning electron microscopy results, looking at those nacreous platelets, there was no statistical difference in condition. Thank you to my volunteers who looked at that. Yeah, no significant difference between the platelets, any level of deterioration. So quick hypothesis check of what we've gone through so far. The first, we thought that abalone raised in high pH water will grow faster. We saw that. Have thicker shells. That was, saw. Develop stronger shells. We saw that. Have a lower magnesium to calcium ratio. We actually saw the opposite of that, where the ones raised in the high pH contain more magnesium than our low, and that they would show less corrosion. And we used our qualitative results here, but again, all of the papers said that there would be differences. We didn't see any differences. Maybe we need to look at it a different way. And finally, that we would see increased dose production in our low pH treatment, and we saw that. So overall, in this system, we saw an 11% increase in abalone growth, comparing our high pH treatment to our low, and increases in shell strength as well, which is huge both for commercial avenues where you would want your abalone in tanks to not be cracking and breaking and getting crushed all the time, and also in restoration. You don't want to get all your abalone ready, put them all out in the wild, and they instantly get crushed up by crabs or stepped on or broken in other ways. We saw a greater level of magnesium in our high pH treatment compared to those other ones. A couple of reasons this could have happened, but one maybe hypothetical is that they were growing faster, and so as they were growing really fast in these really happy conditions, they could have perhaps just been pulling everything they could out of the water in order to grow their shells as fast as possible while they were in these good conditions. When we looked at abalone length, in a lot of the results that I had, the ambient and the high pH treatments were often right neck and neck. This might be due, maybe there really is no difference between the high and the ambient pH treatments. However, there could be an effect kind of through our experimental design. In order to keep our ambient treatment at ambient, we had to bubble in a little bit of CO2 in order to depress the pH that was being raised by the dulse. And so in doing so, if this affected the nutritional profile of the dulse, you could have been giving them happier, healthier dulse, which could in turn look like larger abalone. So a little bit of ocean acidification for your seaweed may not be a terrible thing. I don't know. And finally, when we looked at our XRF and our EDX results, they both gave us statistically similar results, which is huge. And they both have their own utility, though, I will say. The XRF, this handheld device that we had, it was great. You could zap a shell in 30 seconds, and it would give you your results. But it was a much more kind of zoomed out look. It did a square centimeter of space, so you couldn't really get in there in detail. Whereas the EDX, you can zoom in thousands of times magnification, looking at these really tiny structures, which was really cool. I wouldn't have seen all those crazy parts of the abalone shell. However, it did take like 3 hours per shell to do all of this, so it's definitely a lot more work. In future directions, I do think it would be important, and we have incorporated this into our studies that we're working on now, looking at putting dulse in the treatment tanks itself to be able to compare directly how the dulse is growing with and without abalone in the tank under all of these various treatments. We wanted to do that for ours, but we just didn't have the space and equipment to do it. And then if we weren't able to do that again, also taking dulse samples to figure out if the dulse in these different treatments has different nutritional profiles, and if that's the reason why the abalone are growing at different rates, or one of the contributing factors. A way to kind of figure out what's going on with the magnesium in these shells could be to individually tag abalone, because right now we have a chunk of abalone that were this size, and then 5 months later they were this size on average, but that doesn't really give you a specific growth rate for each of these animals. And so if you tag all these animals, regardless of the treatment they're in, if one of them grows really big and the small, you can look at the magnesium content at the beginning and then at the end to see if just increased growth rate itself is what's adding that magnesium into the shell, regardless of treatment. As I kind of mentioned with the scanning electron microscopy analysis, looking at the pictures of those nacreous platelets, we did qualitative because I just wanted to get a general idea if there were any trends to dig into. Did not see any trends. However, there may be some need to develop some sort of AI tool to look at and quantify. I don't know how you would go about looking at that picture and measuring all of those little ones, maybe on ImageJ, but I feel like that would be the worst thing ever. So definitely some smarter tools to look at that. And then finally, I really do think that IMTA with abalone and calcifiers, or with seaweed and calcifiers, has pretty great potential. Currently, it's not used at any farms, mainly because we haven't figured out a really good way to do it. In this system specifically, in these tanks, we're really prioritizing the dulse growth as it needs all that space to tumble through. So I think it would be fun to kind of work with the farm to figure out a way that we could increase the surface area on the tanks while still having our dulse and not getting caught up on the sides of the tanks in order so that we could do this IMTA at a commercially relevant density. The overall utility of IMTA is pretty fantastic as well. In the 5 months that I had these tanks running, due to the tumble culture nature of the tanks and the dulse kind of scraping on the sides, I didn't have to siphon a tank once. Normally a task that would happen at least once a week, so that saved, I don't even know how many hours of work for me there. And then if you're on a farm doing this, spread out through many more tanks, that decreases the overall labor that you're needing. It decreases your feed need, as you are growing all of your algae on site and feeding it directly to your abalone without having to move anything. And it potentially decreases the facilities required as well, as you're growing your abalone and your seaweed in one tank. In a concurrent study, we found out that we could indeed grow our abalone to market size, which is huge, so they can stay in this system and get to a marketable size. And then with using the kind of novel aspect of this design, I think it could really promote selling abalone as the effluent produced from this tank, and this flow-through design has increased oxygen and increased pH, and the nutrients are being pulled out of it, where in a lot of fish farms there was a big problem behind low oxygen and nutrients being released into the wild. I just have like a better look on this already pretty low trophically affected aquaculture operation. And I do really think that there are potentials for other species. We're right now figuring out if we can work with white abalone and do an IMTA with them to produce bigger, faster, and that's going right out directly into the ocean, so that would be huge. And then looking into other species as well, like oysters, to see how we can use this on different calcifying organisms to better their growth. And with that, that's it. I'd like to thank everyone who helped me, my advisors, Maya, Scott, Luke. Thank you so much for all your guidance and everything. Ivano, thank you for not being on my committee, but giving me all the advice and the resources that I needed. I really appreciate that. Rachel Crane at Hopkins, thank you for all the work you did helping us every single day with the materials testing. Andrew Kim, my boy, thank you for teaching me the ways of the lab, ins and outs, goods and bads and bads. Thank you to Monterey Abalone Company for supplying me with all the abalone I needed for my study, and Monterey Bay seaweeds for all the dulse I used in my project, and my funding sources, Sea Grant and Coast. And thank you to all my friends, everyone who has helped me with my project, sampling stuff, inviting me to things. I'm not the one to go out and do stuff, but thank you for having me tag along. Definitely look deep at all those pictures there. There's some gold up there. Everyone who's got me into aquaculture and got me into marine science and pushed me further into that, thank you. And finally, to my family, thank you guys for being here today. It means a lot. Everyone online, thanks for watching. I love you guys. I couldn't have done it without you. Thanks for pushing me to do this, which leads me to my last slide. Thanks, buddy. I wasn't going to do grad school, and then as soon as Steph finished her PT degree, she's like, all right, so where are you going to school? And I'm like, I'm not going to go to school. I don't need to go to school. And she's like, we're going to school. Fast forward, here we are, and now I'm like infinitely grateful that you made me do it. It's the best decision you've ever made. And with that, thank you everyone for being here today, and I'll take questions.
All right, who has questions for Noah? And when you ask your question, you have to use this microphone so that people online can hear you ask the question.
Or not.
Come on, hard questions.
I'm
All right,
on
we'll
the-
go down the line here. Warmed up.
Warmed up. Okay, great job, Noah. That was really exciting to learn everything. You've done so much. And I'm going to ask one, no worries if you didn't, but I'm curious whether you saw any differences spatially on the shells in terms of, say, the calcium-magnesium ratios or anything about the structure, like closer to the growing zones, older parts of the shell.
So when we did our analysis, that was at the very end of the project, our abalone had doubled in size, and so we made sure that you can kind of see on the abalone shell, there's like a stress event from when we counted them and put them into our system, and there's like a little line on the shell. All of our analysis that we did was past that, so we made sure that it was shell that had been accreted down after they had been in our treatments. Looking throughout the shell itself, with the XRF, it was kind of zap the back, zap the front. That's what it is. We can kind of see, and we tried to position it so that it was further along and not back at the apex of the shell to capture that growth. And I mean, there was a trend, so I mean, it must have done something well. But then under the EDX, I looked at several locations on the shell, 2 on the dorsal side, 2 on the ventral side, and then in that aragonite and calcite layer. And that was really interesting because between the different layers of the shell, there was almost no magnesium found in the aragonite layer itself when I looked at it from a cross-section, but when I looked at the ventral surface of it, which is just piled up on top of one another, we saw magnesium there. So there's a lot more that needs to be done, looked at with like mapping out the magnesium inclusions throughout the whole shell. But yeah, it was really bizarre.
Yeah, that's really fascinating, and what it could mean for the structural integrity,
Yeah, and
especially
then-
as the shell ages.
Right, and what's going to happen to that shell. I meant to say something about that, but I forgot, but it's okay.
You just did.
Um,
That's great.
We don't know how the shift in magnesium is going to affect them later in life. Traditionally, magnesium calcite, that inclusion into their shell, is more soluble in water. But hopefully we've kind of got them past that small juvenile stage, shepherded through them. And so now that they're a little bit bigger, maybe they'll be a little more resilient to any changes in ocean acidification from here on out.
Thank you.
Yeah.
Nice job, Noah. I have a question for you about your dulse growth. So usually nitrogen's a pretty important part of vegetative growth, but it looked like with your multivariate stats, they pretty much all showed a negative interaction between nitrate and dulse growth.
Yeah. Yeah.
What's up with that?
Weird, right? So I too was like, what the heck? This is really weird. This was all taken from the seawater sensor itself down in the pump house, so it's Steve Cunningham's fault. But no. So I thought it was going to be like just maybe something weird like that. The sampling wasn't working, but then it was like too strong correlated every single time. And so I read papers, which I probably should have done in the first place. And it turns out that nitrate isn't something that causes the dulse to increase in growth, but it changes its the nutrients incorporated into the dulse. So if you're fertilizing your seaweed to get it to grow more, don't do it with nitrate. Yeah.
So follow-up question. Do you have concerns with using abalone as sort of a fertilizer source, but their waste being converted too quickly to nitrate and then not being available for the dulse?
Yeah, and I mean, hopefully with having them in the same tank like this, it really cuts down on the residency time, and so hopefully there would still be ammonia, their excretes in the water for the dulse to uptake. In one of the other studies that we did, we lowered the residency time in the tank, and it didn't seem, at that flow rate, it kind of seemed like the water coming into the system was the real big thing fueling the dulse growth. But yeah, it's really hard. We have to take like a bunch more samples. We took samples of the dulse to see kind of what the nutrition profile is looking like, but we haven't analyzed them yet.
Coming way to the back here.
Great job, Noah.
Thanks.
Can I ask two brief ones?
All right.
All right, so first one, did you have any concerns at any point about having a high biomass of dulse resulting in a drop in pH at night during the dark cycle?
I'm going to stop you there and answer that, or else I'm going to forget.
Get it?
Yes, that was like a huge major concern. Fortunately, just the aeration, because I mean, that dulse is like, if you've seen the tumble tanks there, it is rocketing around in there. There's lots of air that's getting blasted into there. And it kind of seemed like that alone, plus the incoming seawater, kind of kept it at a happy medium there. And so those graphs that I showed at the beginning, that was over 2 weeks, and it never dropped like way down. It kind of seemed, more so during the day we saw the products of photosynthesis, but we didn't really see the products of respiration just due to the nature of the system.
Cool. This second question is about body size. So you showed those cool time series noting the increase in spread of the distribution of body size. And I was curious if you had, in your analysis, treated body size as a continuous covariate, so that, like, were there consistent effects of body size on any of the metrics you looked at?
And like when that became?
Yeah, like was there, growth declined significantly as a function of increasing body size, for example?
No, I did not look at that, but that is a great thing to look at. I will come and find you to figure out how to do that.
Come on by.
Something for the publication. All right, questions. There's a lot of you in here. You guys got to, he didn't answer every question about abalone. Oh, there we go.
Great presentation. Thank you, Noah.
Thanks, Tom.
So I was curious about the SEM results you had for the, sorry, is it Natra?
The nacreous layer.
Nacreous layer?
Spoken like a fish guy.
I don't know, dude. I barely understand what a snail is in the first place. So I was wondering, your assumption there would be that the low pH in the water would be dissolving the crystals in the nacreous layer, right? But does the snail cover the nacreous layer? Does it like attach to it? Is there like a bodily fluid that has was regulated by the snail that would have a consistent pH covering that material?
Yes. Next. Yeah, so there is part of the snail's body, its mantle covers that, but it's not like a firm attachment like skin covers my bones. It's like I can like move on and off, so there is some interaction there potentially between the seawater and the snail shell.
No. I was wondering, did you look at the dorsal side of the shell and see if that's being corroded more?
Oh yeah. Yeah, I looked at that as well. Again, it's one of those things where it's like a qualitative measure that you have to look at quantitatively, and there's just, you zoom into anything 2,000 times, and like the tiniest little bump is just a massive mountainscape. And so it's really hard to quantify that entire snail shell, and do we look at the edge, do we look at the beginning, and that's why we looked at the underside right in that new nacreous accretion, because that was a spot we could zero in on. But yeah, on the backside of the shell, anything from things growing on the shell to getting bumped while we're sampling, like the tiniest little thing. So yeah, it's really hard to look at that, and I don't have a great way to analyze those super zoomed in pictures quantitatively. If you want to come up with one though, let me know.
Thank you.
Great job, Noah. Killed it. So I'm interested in some of your results. So the first ones, you were saying the ambient seems to do a lot better. I think it was maybe for the growth rates or something. And then later the high, or did I just switch those?
Yeah.
So why do you think like some of the metrics are doing better in high and some are doing better in ambient?
I think it's kind of a crapshoot between the two of them, at least how we have it. Like I kind of said, in our ambient pH treatment, there was a little burst of CO2 added in there to just keep it within the bounds that we had set, and that could potentially be getting it going. And like getting it to the point where it's, even though the pH may not be the best, they're just getting a better quality of food, so maybe that's letting them grow on par with our abalone in the high pH treatment that have that boosted extra pH, but maybe their food isn't as good, and so it kind of maybe cancels out. I don't know. We need to take seaweed samples to figure some of this out. Lots of questions around it.
Thank you.
I didn't know anyone in the phycology lab though, so I couldn't.
Sure.
Great job, Noah. I was curious, you mentioned that the it was an acronym, Integrated IMTA system has been around for a couple decades. Has it been done with, so you focused on red abalone. I didn't know if it's been done for black abalone, or like there are a lot of other species of abalone.
Yeah, so IMTA is not, sorry, was that your question?
Oh, you're good. No,
Okay.
please.
How's it?
That was the question.
It did not start with abalone. It has been around since like the 70s. And people do it with fish and seaweed and worms and all kinds of critters. It's really cool to see. It's not specific to red abalone or abalone in general. We're trying to figure out if we can do it with other things. You should talk to Lucy over there about her stuff. She's looking at oysters and seaweed. We are trying it with white abalone as well. But yeah, a lot of the other studies that I looked at used different species of abalone, the European one, and other species. So yeah, it's been done a lot all around the place, but this specific question for this specific species fueling our California aquaculture, yeah.
Very cool. Thank you.
Thanks.
I have a couple of questions online.
Dun, dun, dun.
Sean Bignami says, great presentation, as usual. Question, does feeding on dulse affect the flavor profile of the abalone compared to traditional kelp-fed abalone?
It's been said that yes, it does. And I think Andrew's told me and Mike said before that it does influence the flavor of it. I have not done a side-by-side, but anecdotally it appears that there is some sort of change to the flavor of them. In the positive, not the negative, yeah. Because you also eat dulse and it tastes like bacon, right, Mike? Yeah, so.
Okay, Aaron says, what shape or structure do shells take on in highly acidic environments? Great job, dude.
Thanks. I would imagine, all of our shells pretty much had the same shape to them. In that one slide that I showed of the oyster, that was a really tiny juvenile that had the big old divot in the side. Mainly, and in a subsequent project that we've done, we've seen in high pH, or low pH rather, they just kind of get a thicker shell. The coloration of the shell can change a little bit, just really brittle and thin, but not so much like you would still recognize it as an abalone shell.
And then Jason Toy asks, did you ever have issues in your higher pH treatments with really low pH water coming in during local upwelling events? If so, did you take any steps to compensate and bump the pH back up, or did you just roll with it?
we just rolled with it. So it was pretty miraculous when I was first trying to figure out how much dulse I needed to throw into the tanks to get a response. I put in it was like no dulse, 1 kilo, 2 kilos, 5 kilos, and then going up from there. And anything past like 2 kilos in the tank, it was all pretty constant. So even throwing just a little bit of dulse into the tank, as long as you got the sun shining, it did a really good job of compensating and bumping up the pH in the tanks. So then we went in through in 10 kilos just to make sure that all bases are covered. We were concerned about the overnight fluctuations, but once again, look at me like that. Wanted to make sure all bases were covered. And then we looked overnight to see if the respiration would take over. Everything looked good. Abalone had plenty of food. Yeah.
All right. With that, let's congratulate Noah. Good job. All right, his committee's going to take him away, interrogate him for a few more minutes, and then we'll bring him back to celebrate with everyone.
Doing great.